Welcome back, my dear students and the friends. In our last lecture, we discussed about the biodiversity conservation. There are two ways of the biodiversity conservation. One is the in-situ conservation that we have discussed in detail. Like in-situ conservation means conservation of the species in its natural habitat, like declaring in areas as a protected areas like national parks, sanctuaries, conservation reserve, community reserve. These are the four categories of the protected areas. Apart from these, we have some centrally sponsored scheme like Project Tiger, Project Elephant. So we had already discussed all these in our last lecture. Now this is the time for to discuss about the ex-situ conservation. So ex-situ conservation is actually the conservation of the species outside its natural habitat by creating the more or less similar habitat for the species to survive. So ex-situ conservation refers to the conservation of the species outside their natural habitat. This approach is employed when in situ, on site, or in the wild conservation measures alone are insufficient to ensure the survival of the species. Ex-situ conservation involves the removal of individual or entire population from their natural habitat and their placement in controlled setting such as zoos, botanical gardens, seed banks, or captive breeding programs. So there are several methods for ex-situ conservation, including there are several methods for the ex-situ conservation. Why ex-situ conservation? Why we need it? Ex-situ conservation plays a pivot role in the biodiversity preservation by providing a safe haven for endangered species. Suppose there are some species whose population is actually very, very low. So in that case, what we do, we bring the population in the captivity and do the conservation, ex-situ conservation, maintaining genetic diversity, critical for species survival and adaptability. Surviving as a vital backup in the face of habitat loss, climate change, and other threats. Serves as a backup for in-situ conservation efforts and facilitating research, education, and awareness on conservation issues. So that is why we need ex-situ conservation. There are different methods of ex-situ conservation: zoos and aquarium, seed banks, botanical garden, gene banks, and cryopreservation. And of course, if you are talking about the zoos and aquarium, we will be discussing about the captive breeding programs. So let's start with the zoos and aquarium. Zoos were actually they started 5,000 years before Christ BC, and basically we used to have the private zoos. People used to collect the animals, bringing animals from different. There were no any law, nothing, nowhere. So that's how the concept of zoos actually started. They did not have recognition. Those who wanted could have keep the animals. So that's how the zoos started. Most of the zoos were the private zoos. So these institutions house and care for a variety of animal species, providing us controlled environment that aim to replicate the natural habitat. Zoos often engage in breeding program also for endangered species to maintain genetic diversity and prevent extinction. If you want to save the species from the extinction and population is really very, very low. So what we do, we bring some viable population to this zoo for the captive breeding programs so that population can increase and we can release the species in the wild. There was amendment in the Wildlife Protection Act 1972. A separate chapter 4A, contains section 38 A to J, was added to the Wildlife Protection Act 1972 for establishment of Central Zoo Authority. Central Zoo Authority is based in Chennai, and that is the controlling over all the recognized zoos, controlling body over the recognized zoos in India. So according to Central Zoo Authority, accordingly, Central Zoo Authority was established as a statutory body under Ministry of Environment and Forest by Government of India in year 1992. So Central Zoo Authority of India is the governing authority of all zoos in India, and is an associate member of the World Association of Zoos and Aquarium. We call it WAZ, World Association of Zoos and Aquarium. For the overall management of the animal house in the Indian zoos, standard and norms of appropriate housing has been laid down under recognition of zoos rule. Because if you are keeping the animal in the captivity in the zoos, we have to follow some rules and regulation. So those rules and regulation were laid down under recognition of zoos rule 1992, which was further amended in the year 2009 and 2013. This is a normal process. We keep on doing the amendment in different rules and acts. So the main function of the zoos are to display, certain and educate the public. That is the main nature, to display, entertain and educate the people, create the awareness among the masses. Very strong emphasis on scientific research and species conservation. Here, rescued animals find refuge in the zoos. Captive breeding programs are also carried out by certain zoos. At present, there are 155 zoos recognized by the Central Zoo Authority. There are many private zoos existing before this rule, before this amendment. They all were the private zoos. So, out of more than 300 zoos, 155 zoos got the recognition, and some small zoos were merged together. So, this was Central Zoo Authority under Section 38H of the Wildlife Protection Act 1972 in the country. The second is the zoos are also involved in the captive breeding program. When the population is actually very low in the wild, what we do, we bring some of the population in the zoos and we do the captive breeding. So definition of the captive breeding is the breeding outside natural habitat, zoos, rehabilitation and breeding centers, botanical garden under observation. The captive breeding program means the breeding taking place under the observation, and this was started 5000 BC in India. So a main objective of the captive breeding is the maintaining the species which are extinct in wild, rescue the imminent danger of extinct, and for conservation related research. So there are different action plan, and these action plan are made by the international bodies. It is not only for India, it is for the world. There are 4 main groups that provide action plan for captive breeding, like IUCN, International Union for Conservation of Nature, CBSG, Captive Breeding Specialist Group of IUCN. WAZ, we have already discussed, that is World Association of Zoos and Aquarium. BGCI, Botanical Garden Conservation International. So these are the organizations, these are the institutions which actually make the action plan for the captive breeding program. There are many criticisms of the captive breeding program. Some say it's good. Being the scientists, we say yes, it is good. But there are also criticism because it's a very, very expensive procedure. It focuses on a few charismatic endangered species, definitely those species which are highlighted, which has been in demand. Those are charismatic, so definitely we are focused on that. Genetic diversity may have sunk too low to be regenerate. Costly, it's a costly effort because bringing the animal, then translocating the animal in the wild, it's a very, very expensive affair. So costly, diverse resources from much more cost-effective ecosystem and habitat conservation measure. Gives false sense that battle against extinction is being won. What if the habitat no longer exists? We are doing captive breeding program, but what if the natural habitat is no longer exists? So these are the criticism of the captive breeding. But so with this, we carry out the captive breeding program guidelines. There are certain guidelines for successful reintroduction after the captive breeding program. When the population increases, we have to reintroduce in its natural habitat. That is the main purpose of the captive breeding program. So for that, we need a self-sustaining captive population, require a suitable amount of adequate and protected habitat where we are going to reintroduce, effective techniques to prepare animal for reintroduction, and post-release monitoring and evaluation. After release of the species in the wild, we have to do the monitoring. We have to keep these species in quarantine for some time. Then only we can release these species in complete wild habitat. There are many successful programs of the captive breeding, many successful. The aim of captive breeding is successful introduction and reintroduction in natural habitat. The aim was archives achieved successfully as well as unsuccessful. In some cases, it was really unsuccessful. So there are international examples of successful reintroduction like red wolf, the first picture itself showing the red wolf, Scimitar horned oryx in Arabia, they are found in the Arabia, and black-footed ferret, California condor, and the Mallorcan midwife toad. So these are some successful programs in the world, and among the plants, St. Helen redwood, round bottle palm, these are the most successful captive breeding program, reintroduction program for some plants also. This is the international. We have captive breeding programs in India. There are many successful programs in India also. Like Central Zoo Authority is the central organization in which actually oversees the captive management program in zoos. Botanical Garden and NBRI, National Botanical Research Institute, is for breeding of endangered plants in Ex-situ. So there are many zoos selected for captive breeding program for threatened animals. Some successful reintroduction are the gharial, pygmy hawks, turtles. So these are some very successful captive breeding programs in India. In order to infuse new technology in the field of reproduction and molecular characterization of endangered species, a laboratory of conservation of endangered species like corns has been established in Hyderabad to take care of the molecular studies. Every species we are protecting, we need to understand the molecular structure of the species. So there are many zoos who are participating and contributing, coordinating the captive breeding program. For example, for the lion, we have captive breeding programs. Coordinator zoo is Junagadh and participatory zoos are the Bhopal, Hyderabad, New Delhi. So captive population is around 82. Snow leopard, that is coordinating zoo is the Darjeeling and participating zoo is Leh, Kufri, Nainital, Gangtok. Clouded leopard, we have captive breeding program. We have captive breeding program for red panda. Coordinating zoo is Darjeeling and it is participating zoo are the Gangtok and Yachuli. Rhinoceros, Guwahati and participating zoo are Patna, Kampu, Delhi. Bro antler deer, which is also called as the Shanghai or dancing deer. The coordinator zoo is Imphal, while the participating zoos are the Guwahati, Kolkata and Delhi. Sun bear, Aswal and participating zoo are the Guwahati. So there are many examples these for the where zoos are involved in the captive breeding programs. So we have some success story. The first success story is the crocodile rehabilitation project. This project, the crocodile conservation project, was launched in 1976, and the main aim of the project was to save India's 3 endangered crocodile species. They were very much threatened because the eggs were removed from its nesting sites, and they were being killed, being poached, and demand is very high in the international market because of this skin. You can see crocodile leather purse, crocodile leather jackets and all. So they are having very high demand. So because of that, they were being poached at different places. So the population got declined. So this project was launched, captive breeding program for crocodile were launched in 1976. And this involved invention measure like captive breeding of a species, and this program also involved the collection of eggs from natural habitats, subsequent hatching and rearing of crocodiles, gharial in captivity to reduce mortality due to natural predator and finally release into the river. So what we did under this program, usually collection of the eggs from the natural habitat, and they were kept in the zoos or the breeding center, and hatching and rearing of the eggs were going on. Hatching and rearing of the crocodile gharial in captivity. It was under the supervision so that the mortality can be reduced. To avoid these threats, this was under the supervision. So there are many areas where these kind of programs are going on. Kukaral, that is in the Lucknow, is one of the most successful breeding center where crocodile breeding center is going on. Patna, Bhitarkanika, Madras Crocodile Bank. So these in Chennai these are the examples where these programs were completed, carried out very, very successfully. The second is the most successful program is the pygmy hawk program, and it was started in 1995 after declining population of a pygmy hawk. Pygmy hawk are actually found in the northeast India. So the pygmy hawk is thought to be the sole host of pygmy hawk sucking louse, Hematopius oliveri, which owing to this status of its host is also accorded in endangered category in the 1994 IUCN Red List of Threatened Species or Threatened Animals. Basista near Guwahati. Basista is a place near Guwahati, is the selected center for captive breeding program. So for the captive breeding program of pygmy hawk, Basista was selected, and some individual release in captive breeding program was carried out in Basista. And when the population increased, some individual were released in Sonai Rupai Wildlife Sanctuary and Manas National Park. The third most successful program was the vulture captive breeding project. Vulture population was decreased. 4 species of vultures. The population declined because of the various reasons. One of the reason is the use of diclofenac in the cattle. So because of the use of the introduction of the diclofenac in 90s, the population, which is a painkiller for the cattle, the population of the vultures declined. After feeding on the dead carcasses, the population started declining, and it was a drastic decline. 90% between there was a drastic decline, almost 90% between 1992 to 2001. The same years in 90s, diclofenac were introduced. So this causes need of captive breeding of vulture species and started from 2001 in Penjore, Raja Bhaktwa and RFR Guwahati. So these are the places where the captive breeding program were introduced, and they were really successful, and population were reintroduced in different area. Started introducing in 2012. Environmental condition were better. So the next is the captive cultivation of the wild plant. 2 species of plants are assessed extinct in wild by IUCN, International Union for Conservation of Nature. Not only the animals get extinct in the wild, but there are plants also which were assessed extinct in wild. Euphorbia maronatheri and Corypha tailera. Their conservation measures are running in Kerala and BRI National Botanical Garden. Many other critical endangered and threatened plants are also cultivated for research in wild. So not only for the animals, but for the plants, captive breeding programs are going on. Then botanical garden. The second ex-situ conservation, important conservation, is the botanical garden. These garden preserves and cultivate a diverse collection of the plant species. They often focus on rare or endangered plants, ensuring their survival and providing a resource for research and education. So like the animals, they also create awareness, because every plant whatever we are getting from the plants, whether it's a food, water, oxygen, whatever you just name, it's all coming from the environment, from the plants, from the biodiversity. We are totally dependent on the natural environment, the natural resources of the environment. So, like the animal, the plants are very, very important. So, in NBRI, two species of plants are assessed extinct in wild by IUCN, that is Euphorbia and Coryphatillia. The conservation measures are running Kerala NBRI National Botanical Garden, and many other critical endangered threatened plants are also cultivated for restoration in wild. So seed banks, seeds are very, very important for growing any crop. Earlier, in the old days, what we used to do, the farmers used to keep, if they are growing the wheat, they used to keep one sack of the seeds for the next crop. They used to, so that's how the seed banks were started. They used to exchange their seeds among themselves, so that on rotation basis they used to keep the best seeds of the crop. So seed banks store seeds from a wide range of plant species, particularly those that are endangered or have economic importance. So seeds are also preserved in the seed banks. Those seeds, those plants which are actually threatened. These repositories help safeguard genetic diversity and can be used for habitat restoration and agriculture purposes. So the Indian seed vault is a secure gene bank located in a high altitude pass on the Changla in Ladakh. So Ladakh is the place where we have one seed bank, seed vault in India. It was built in 2010 jointly by the Defense Institute of High Altitude Research and National Bureau of Plant Genetic Resources, and it is second largest seed bank in India. Till date, Navdanya has set up 150 community seed banks in 22 states of India in last 30 years. Navdanya is a non-government organization running by the very renowned environmentalist Miss Vandana Shiva. So she is heading the Navdanya, and they are basically working on the indigenous knowledge, seed preservation and all. So the last, the ex-situ conservation is the cryopreservation. There are many examples, there are many things under the ex-situ conservation. So one of the example of the ex-situ conservation is the cryopreservation. Some conservation efforts involve the preservation of genetic material such as sperms, eggs or embryo through cryopreservation. Freezing of the genetic material, as simple as this. So this method allows for the storage of genetic material for future use such as artificial insemination or in vitro fertilization. So cryopreservation derived from the Greek word cryo meaning cold. So freezing the genetic material in a very, very cold environment. Thus it refers to the preservation of biological tissues in sub-zero temperature, typically minus 196 degrees centigrade. So plasma preservation is minus 18 degrees centigrade, but the cryopreservation is in minus 196 degree centigrade. At these temperature, all biological temperature is presumed to provide an indefinite, if it not near to infinite longevity to the cells. Okay. Pre-implantation embryos, oocytes, spermatozoa, ovarian tissues can be cryopreserved. So cryopreservation means freezing of the genetic material for the future use under minus 196 degree centigrade. There are various benefits of the cryopreservation. Effective means to conserve the germ plasm of endangered species. Fertility preservation method to reduce multiple pregnancy. Large range of stock available, easy disease-free exchange of stocks nationally and internationally, and safety from disease, genetic contamination and breeding failure. The latest, the best example is of Mona Singh, the TV actress and the film actress. She is the popular actress known for her role in TV serial Jassi Jassi Koi Nahi. everybody knows Mona Singh, and she was very popular after the TV serial Jassi Jaisi Koi Nahi, and film like Three Idiots and Son of Sardar. Opted for ex freezing at the age of 34. She kept her eggs for freezing at the age of 34 because it says up to certain age the eggs are very fertile. After that they start deteriorating. Usually in medical term they say after 35 the eggs start deteriorating. So they work but not as effective as before 34. So she froze her eggs at the age of 34 and she expressed her desire to have children. She expressed her desire to have children, but also acknowledged the importance of pursuing her career goal without feeling uh, pressured by social expectation. So definitely, so she is not she, but many celebrities, many people are keeping these eggs and also germ plasm. So, ex-situ conservation is a vital part of global strategy of preserve biodiversity. By understanding its benefits, challenges, and the importance of ethical consideration, we can enhance our conservation effort and ensure the survival of a species for future generation. The genes, the germ plasm of cheetah is also preserved in the Lacons. Lacons is the laboratory for the endangered species in Hyderabad. So people are trying to regenerate or to develop the cheetah in the laboratory. So if we can do it wisely, if we are successful in many cases. If we will be more successful, we can revive many species. So this is all about today's lecture. I hope you enjoyed my lecture, especially the Mona Singh example. Thank you.